special senses. We're going to be talking primarily about the senses that are housed in the eye and the senses that are housed in the ear. So we'll start with the eye. The eyes use photoreceptors. So these are photoreceptors, that's the modality. They're exteroreceptors, getting information from the outside world, and it's a special sense because it's housed in the sense organ in the head. So eyes are using photoreceptor cells to help form visual images of the environment. The eyes have many accessory structures that help to protect the eye against foreign objects and to ensure that the surface of the eye remains clean and moist. So some of those accessory structures are things that you're familiar with, things like the eyebrow and the eyelashes are hairs that prevent things from falling into the eye. The eyelids maintain the moisture of the eye. The conjunctiva is the mucous membrane that covers the inside of the eyelids and the surface of the eye. So if you were to look at the inside surface of the eyelids, you see all those tiny little blood vessels. Those same blood vessels travel over the surface of the eye in the white of the eye. Now, usually you don't see the conjunctiva, those blood vessels that are traveling over the white of the eye, but you do when somebody's eyes are bloodshot. And this is when there are tiny little blood vessels in the conjunctiva and they fill up with pooled blood. And this can happen for various reasons. It can be irritation, allergies, dehydration, low blood pressure. One case that you may have seen where people get red bloodshot eyes is after consuming cannabis. And the reason that you get bloodshot eyes after consuming cannabis also happens to be the same reason that cannabis is a really excellent treatment for glaucoma. So some of the cannabinoids within cannabis, THC, lowers blood pressure. That lowered blood pressure in turn causes blood vessels and these very small capillaries to dilate. When these capillaries dilate, it increases blood flow to the eyes. It reduces intraocular pressure, which is good for glaucoma. But that increased blood flow and pooling of blood within the capillaries causes redness. So the decrease in pressure is that exact effect that benefits glaucoma patients. And this is because if you're smoking, it's not the smoke that's making your eyes red, it's the cannabinoids. And that'd be affecting the conjunctiva that lines the inside of the eyelids as well as over the white of the eye. Other accessory structures include the lacrimal apparatus, and that's shown down below on this slide. Remember that when we were looking at the bones, we had two separate lacrimal fossa, one in the frontal bone where the lacrimal gland is located. So this is where tears are produced, and tears are produced and secreted out through the lacrimal ducts, and they travel medially and inferiorly over the surface of the eye, and then they get collected at the lacrimal canals and drain into that lacrimal fossa at the lacrimal bone. So all of these are accessory structures that are helping to protect the eye, making sure that it's remaining clean and moist. So the anatomy of the eye, the wall of the eye is constructed of three different tunics. So the tunics are the layers of the eye, and they fall into three different categories. The outermost is the fibrous tunic. The fibrous tunic is tough, it's supporting the outside of the eye, and it's composed of two different regions. The first one is the sclera, and that's the majority of the fibrous tunic. So the sclera is all the way around the entire surface of the eye, except in the front. So it makes up the majority of the fibrous tunic. It's the white of the eye, and it's providing shape, it's protecting the internal parts of the eye, and it's made of dense, irregular connective tissue. The fibrous tunic, like the other tunics, has anterior modifications to make way for the pathway for light. So the sclera doesn't let light pass through it, it's the white. So the anterior modification of the fibrous tunic is the cornea. The cornea is transparent and it's avascular. It's getting oxygen and nutrients from the lacrimal fluid flowing over the surface of the eye, as well as from the fluid that's inside of the eye. So the cornea is transparent. It allows light to pass through. That means it's avascular. The next tunic is the vascular tunic, and it's composed of 3 different regions, from posterior to anterior. The posterior, which is lining most of the back of the eye, this middle layer is vascular tunic, that's the choroid. And the choroid has this vast network of capillaries supplying nutrients and oxygen to the retina. The next part of the vascular tunic is the ciliary body. The ciliary body is composed of the ciliary muscles, and it's responsible for changing the shape of the lens. The last part of the vascular tunic is the iris, and the iris is the pigmented part of the anterior eye. When people say that they have brown eyes, green eyes, blue eyes, they're talking about the iris, and that's part of the vascular tunic. And the iris controls the amount of light that's coming into the eye. So the vascular tunic is the choroid, this vascular layer, plus the ciliary body, which changes the shape of the lens, and the iris, which changes how much light is entering into the eye. So it's a sphincter muscle. The last tunic is the retina. The retina is this internal layer of the eye, the innermost layer. And it consists of 2 layers. There's a pigmented layer, and that's attached to the choroid. That pigmented layer absorbs any light that passes through the retina. And then the innermost layer is the neural layer. And this is where those photoreceptor cells and associated neurons are. It's receiving light and converting energy into nerve impulses. And one thing that I didn't talk about here is this structure. This is the lens. The lens isn't considered to be a part of any of these 3 tunics, but it's most closely associated with the vascular tunic. So we said that the iris, part of that vascular tunic, it's that part with color. So in this case, this person has a blue iris, indicating a lack of melanin in the iris. So the iris is the part with the color, and it's a sphincter muscle that controls the amount of light that's coming into the eye. The pupil isn't actually a structure, it's just a hole, and it's this black hole located in the center of the iris of the eye, and it's what allows light to strike the retina. So it appears black because light rays that enter through that hole, that enter through the pupil, are either absorbed by the tissues inside of the eye directly, or they bounce around a little bit and are absorbed by the tissues inside of the eye. You all probably know that the pupil gets wider in the dark and narrower in the light, and that's what's shown here. In bright light, the pupil constricts to allow less light to strike the retina. And this is when the sphincter pupillae muscle constricts. When a sphincter muscle contracts, then it reduces the size of an opening. And this occurs under parasympathetic innervation or under bright light conditions. Under low light conditions, or with sympathetic innervation, that is that fight or flight response, then the dilator pupillae contracts, and that expands the size of the pupil. So that causes the pupils to dilate. And you don't need to know the names of these 2 muscles, just know that the iris is this sphincter muscle that controls the diameter of the pupil, and thereby the amount of light that's striking the retina. So we've got another view of the anatomy of the internal eye. We already talked about the 3 tunics. That is the outermost fibrous tunic with the sclera, the white of the eye, and then out here, this transparent modification of the fibrous tunic, the cornea, that transparent avascular layer. Deep to that is the vascular layer, the vascular tunic with the choroid, that highly vascular layer that's supplying the retina. And the ciliary body that's responsible for changing the shape of the lens, as well as the iris that's responsible for changing the size of the pupil. And then that innermost layer, the retina, which is the pigmented layer, and the neural layer on top of it. One thing that we haven't yet talked about is the chambers of the eye. So the eye has a couple of different chambers. There's up here, this whole area between the cornea and the lens. This is the anterior cavity. And then everything between the lens and the back of the eye, that's the posterior cavity. And they contain different fluids. So between the cornea and the lens in the anterior cavity, there's aqueous humor. This is watery fluid that's serving the cornea, that's serving the iris, where there's no blood flow in the cornea here. And the anterior cavity is divided up into the anterior chamber, which is between the cornea and the iris, and the posterior chamber, which is between the iris and the lens. So you would say that the anterior chamber of the anterior cavity contains aqueous humor. The posterior chamber of the anterior cavity also contains aqueous humor. Behind the lens, in the posterior cavity, this is where you'd find vitreous humor. Vitreous humor is much more like jelly, and it's what helps to support the eye. And this is something that you'll see on the eye dissection. So let's take a look at that eye dissection. Typically, we do a cow eye dissection in class. Unfortunately, we're not in class, so I found a brief video that shows the cow eye dissection.
Welcome to the 3-minute eyeball dissection. Make sure you grab some safety glasses and glove up. Let's get started. First, I want to show you the tools we'll be using. Dissection scissors and a blunt probe, that's all you'll need. You'll notice the eyeball I have here is nice and clean and smooth. Well, that's because I removed all of this connective tissue, fatty tissue, muscle before I got started here to save us just a little bit of time. If you're doing this dissection in lab, you're probably going to have to remove a little tissue before you begin. We set the camera down and let's get started. All right, on the outside of the eye, we need to identify a few surfaces. All of this area out here around the circumference of the eye is the sclera. In front, we have the cornea. That's actually where we're going to make our first incision. It's going to be right in between the cornea and the sclera. Before we check that out, let's flip the eyeball over to the back. And let's make sure we notice this right here, which is the optic nerve, the remnant of the optic nerve. So make sure you check that out. All right, our first incision is going to be between the cornea and the sclera. We need to gently puncture the eyeball. Now be aware the contents of the eyeball are under pressure. In the front, there is the aqueous humor. In the back of the eye, there is the vitreous humor. These are under pressure. If you pop this and squeeze the eye, it will spray humor everywhere. Trust me, there's nothing funny about getting covered with humor. All right, do this very carefully. Be careful not to stab yourself or anyone around you. There we go. Safely puncture the eyeball. Let me complete this incision around the cornea. All right, we're going to flip this up. Right here attached to the underside of the cornea, put this back here, is the iris muscle. That's what this black structure, this black ring right here is. Right in the middle is the pupil, and right behind that is the lens. Now, if this animal were alive, the lens would be extremely flexible, but now that this is preserved, it's extremely hard, almost like a marble. Very cool to take a look at. This is the rest of the eye. Notice it is full of this jelly-like vitreous humor. We need to squeeze some of that out before we can continue with our cut. Vitreous humor. All right, we're going to split the eyeball in half at this point. You want to do this carefully. Miss the optic nerve there. Be very careful to keep your fingers out of the way. If you're doing this on your own, take your time. You do not need to do it this quickly. I'm a trained professional. On the inside of the eye, you'll notice this first layer right here, this light layer. That is the retina. That's what actually allows you to see. It contains those photoreceptors, the rods and cones. We need to get rid of the retina to see the layer underneath, and that is the choroid. In humans, the choroid is just like this up here. It's black. But in many types of animals, including cows, they have an additional reflective element of the choroid called the tapetum lucidum that allows them to see in low light conditions at night. It's also what causes eye shine in many types of animals, including cows. If you look at the retina right behind the optic nerve's right here, and then just inside of here, opposite the optic nerve, you will see the blind spot. That's where the retina attaches to the optic nerve. All right, those are our structures for the eye. I hope you learned something. Join us again for more dissections.
There will be questions on the exam related to cow eye dissection, or you'll have to identify structures from pictures. So make sure that you take a look at that. So let's get back into it. So he was just telling you about the retina, that layer that contains the photoreceptors. There are two features to be able to identify on the retina, and that's the optic disc and the fovea centralis. So over here is illustrated the fovea centralis and the optic disc. And then over here is a simulation of what it would look like if you got a photograph taken of your retina. There's the fovea centralis, and then here's the optic disc where the optic nerve is leaving the eye. So the fovea centralis, it's a depression, a little pit in the retina. It contains the highest proportion of cones that are responsible for color vision and almost no rods. It's responsible for really sharp central vision, which is essential for us to do activities where you really need those visual cues like reading, driving. You're going to look at something that's directly in front of you. That's where you're going to have the clearest, sharpest vision because of this fovea centralis. The optic disc is a blind spot on the retina, and this is a spot where there are no photoreceptors. And this is where neuron cell axons are exiting the retina to form the optic nerve. That's cranial nerve number 2. And this is also where blood vessels enter and exit the retina. So at this optic disc, there are no photoreceptors, so there's no neural layer to the retina right here. So this is a blind spot in your vision because there are no photoreceptors at the back of the retina at the optic disc. And it's going to appear as this more yellow looking structure on a picture of the retina. And I'm going to show you a demonstration of the blind spot or the optic disc. Special senses are a great time to learn some good party tricks. One of them is finding your blind spot. Everybody's got blind spots, and a good way to demonstrate this is by making a card that looks just like this. You can use any piece of paper. I got an index card, and on it I've put a plus sign and a circle. And what I'm going to do is look at it with just one eye. So I'm going to look at it with just my right eye, close my left eye, and I'm going to look at the plus sign, and I'll slowly move it away from and toward my face. And at some point, this circle will completely disappear. I won't be able to see it. It'll be in my blind spot. So I'm covering up my left eye, and I'm looking at the index card, the plus sign, with my right eye, and moving it slowly toward myself. And right there, I can see just a white index card with a plus sign on it. I can't see the circle at all. This is where the circle has fallen on my blind spot, also known as my optic disc, where that optic nerve, cranial nerve number 2, is leaving the retina. So there are no photoreceptor cells in that spot. So you can amaze anybody that you're sheltering in place with by showing them this trick All you need is a piece of paper with 2 marks on it. Special senses are a great. So let's get back into the science, and we can leave the party tricks behind for now. So the structure and organization of the retina, I've been talking a lot about those photoreceptor cells. Those photoreceptor cells are rods and cones, and they're called this just because that's their shape, that's what they look like. Rods are responsible for vision in dim light and your peripheral vision. They can detect movement. And then cones are important for precise vision and color vision. So they both have their roles to play. So we're looking at a section right now of the back of the eye. All of this, here's the pigmented layer of the retina, and here's the neural layer of the retina. And deep to that is the choroid. Out here would be the sclera. So we're following with this yellow arrow the path of incoming light. And incoming light strikes the retina, and it passes through some layers of neurons that will end up processing and sending that information. So it passes through ganglion cells, it passes through bipolar cells, and then it strikes and stimulates these photoreceptor cells at the back here. So the rods and the cones are stimulated by that light, and any excess light is absorbed by the pigmented layer at the back of the retina here. So it's these cones and rods that respond to the incoming light, and then that information gets passed to the bipolar cells. Remember, these are the specialized type of neuron that only found in sensory structures. It gets passed to the bipolar cells, and then it gets passed to the ganglion cells. And the ganglion cells, their axons, is what makes up optic nerve, cranial nerve number 2, that's leaving the eye at the optic disc. So light passes through the neural layer and is absorbed by the photoreceptor cells. This causes a conformation change in the photoreceptor cells, and then causes an action potential to be passed through the bipolar cells, through the ganglion cells. So there are those 3 distinct layers that are forming the neural layer of the retina. The photoreceptor cells, that's that outermost layer composed of rods and cones. The rods are really important in dim light and for peripheral vision. And the cones are what's important for precise vision and for color vision. When those are stimulated by light, then they send a signal to the bipolar cells. The bipolar cells synapse with the photoreceptors and with the ganglion cells. And then the ganglion cells are the innermost layer of the retina. The axons of these cells leave the retina, and they form that optic nerve, cranial nerve number 2. If you look back at the image of the neural layer, you'll see that there are some additional neuron types in there. There are horizontal cells between the photoreceptor cells and the bipolar cells, and there are amacrine cells between the bipolar cells and the ganglion cells. And this is going to be important because there are a lot more photoreceptor cells than there are bipolar cells, and a lot more bipolar cells than there are ganglion cells. So these are helping to integrate that message to do this neuronal convergence. So they're helping to process and integrate that visual information. So now that we've been talking about the retina for a little while, hopefully you can answer this question. Which of these statements about the photoreceptors of the human retina is false? Let's talk through it. So part A, cone cells and rod cells differ in shape. That's true. One looks like a cone, one looks like a rod. So that's not the correct answer. B, rod cells are highly sensitive to light. That's true. That's how they operate under really low light conditions. C, cone cells provide high acuity color vision. That's true, you know, they're responsible for crisp vision and color vision. D, the density of rods and cones varies across the retina. That's true too, and I'm about to show a demonstration of how you can test that. Part E, cones outnumber rods in the retina. That's the one that's false. So it's primarily rods throughout the retina. You primarily have that low quality gray vision. And then in areas of high acuity, like the fovea centralis, this is where cones would outnumber rods. But across the whole retina, it's mostly rods. And I'll show a demonstration of this that you can try at home. I'm going to show you a cool party trick that helps to demonstrate some features about the distribution of the two types of photoreceptors, rods and cones, on the retina. And you can do this with anybody that you're sheltering in place with to amaze them. So what I'm going to do is I have this box of a bunch of different colored pencils, and I'm going to take one of them out at random, and I'm going to ask my volunteer to look straight ahead and keep looking straight ahead. And I'm going to move one of these colored pencils into his peripheral vision. I'm going to ask him to raise his hand when he can see the colored pencil in his peripheral vision. And then I'm going to have him guess what color it is. Okay, you ready?
Yes.
Okay. I'm going to move it very slowly.
All right.
Okay, what color is it?
Red.
Incorrect.
Wow.
It's celery. You want to try another one?
Oh, sure.
Okay. Look forward again.
This is Marley, my assistant. He's helping.
Look forward. Okay. You ready?
Yes.
Okay. I'm going to move it very slowly. What color is it?
Orange.
That's close. It's yellow.
Oh, wow.
What was that demonstrating, Matt?
Eye anatomy, retinal anatomy.
Okay, where is the highest concentration of cones, those photoreceptors for color in the eye? The fovea, and that's in the front here. When stuff's in your peripheral vision, it's detected by rods. Those are good in low light conditions, but they're not so good for color. So you can try this at home. Sheltering in place is going great. Day 6. Okay, let's get into some more features of the eye. The lens, I said it's not really one of the 3 tunics, but it's most closely associated with the vascular tunic. It's transparent, it lets light through, and it can change shape. It's deformable. It's held behind the pupil by the suspensory ligaments. The suspensory ligaments attach to the outer capsule of the lens, and when these tense or slacken, then it changes the shape of the lens. These changes in tension are caused by the contraction and relaxation of the ciliary muscles, part of that ciliary body. So when you're looking at something far away, like a landscape, then the ciliary muscles are relaxed, the suspensory ligaments are taut, and the lens is flattened. To accommodate or to look at something close, like your watch, then what happens is that parasympathetic fibers excite the ciliary muscle contracts. This causes the suspensory ligaments to relax, and the lens becomes more spherical, and that allows you to look at something that's close. So the lens is thickened and more spherical. This is why your eyes get tired when you stare at something that's too close, like a computer screen or something under the microscope for too long, because looking at something close requires contraction of the ciliary muscles and they get tired. Looking at distant things doesn't involve this muscle contraction. The last thing to talk about are the visual pathways. So you know that light is striking the retina at the back of the eye and being processed by those photoreceptor cells, passed to the bipolar cells, passed to the ganglion cells. And the ganglion cells exit the back of the retina at the optic disc and form the optic nerve. And then they travel back here to the optic chiasm. This X marks the spot in the diencephalon. And at the optic chiasm, we get optic nerve axons from the medial region of the retina crossing over, and some of them don't cross over. They remain uncrossed. So at the optic chiasm, you can get information from the left eye being passed to the right side of the brain, and information from the right eye being passed to the left side of the brain. And this is occurring at that X marks the spot, the optic chiasm. This means that on either side of the brain, there's going to be information about the left and the right side. So ipsilateral information and contralateral information. And that allows you to build a fuller picture of the world. So this optic tract continues back to the thalamus or the midbrain, this relay point, and then all the way back to the primary visual cortex of the occipital lobe, getting that processed information from the thalamus. So this occipital lobe, that's that primary visual cortex. So now we're going to switch gears and get into some of my favorite anatomical structures and some of the most complex in the body, as well as some of the smallest. And these are all of the structures that are housed within the inner ear and that are responsible for your senses of equilibrium and your sense of hearing. So the ear is divided into these three distinct anatomical regions. The external ear, and this is what you see, what you're familiar with. The external ear is this skin-covered, funnel-shaped, elastic cartilage-supported structure that's called the auricle, A-U-R-I-C-L-E, or sometimes called the pinna, P-I-N-N-A. And the auricle leads to a bony tube that you know about in the temporal bone. That's the external acoustic meatus. And the external ear ends at the tympanic membrane, also known as the eardrum. So this tympanic membrane marks the border between the external ear and the middle ear, the next compartment of the ear. The middle ear is an air-filled cavity called the tympanic cavity. Here's the tympanic cavity right here. And this is just medial to the tympanic membrane. And that has the three auditory ossicles that we talked about when we talked about the bones. So we got the malleus, incus, and stapes, or hammer, anvil, and stirrup. One feature of the middle ear is this that's called the auditory tube, or the eustachian tube, or sometimes called the nasopharyngeal tube. The auditory tube is the simplest name for it. And this auditory tube connects the nasopharynx, which is part of your respiratory system, with the tympanic cavity of the ear. So it connects this air-filled cavity with another air-filled space. And you're familiar with your auditory tube because whenever you feel like you need to pop your ears, what you're doing is you're forcing air through that auditory tube. This is important because for the ear to be able to work properly, you need to have equal air pressure inside of the tympanic cavity as well as outside in the external acoustic meatus. And that's what allows for the tympanic membrane to vibrate back and forth and to transmit sound. If air pressure builds up in this tympanic cavity, then it's pressing outward on the tympanic membrane, and that means the tympanic membrane can't vibrate as well, can't transmit sounds as well. So when you pop your ears, what you're doing is equalizing the pressure on either side of the tympanic membrane and allowing it to vibrate freely when it's struck by sound waves. And then we'll take a deep dive into the inner ear in just a second. But while we're here, I'll show you that it's the semicircular canals. There's 3 of them. These are responsible for dynamic equilibrium, so knowing when your head is spinning. There's the vestibule, which contains your sense of static equilibrium, or tells you, that your head is tilted in one particular direction, and the spiral-shaped cochlea, which contains the organ of hearing. And here is the vestibulocochlear nerve connected to the vestibule and to the cochlea. These branches merge to make cranial nerve 8, and that travels through the internal acoustic meatus. So all of this is going to be housed in the petrous portion of the temporal bone. So you're all familiar with the external ear. Let's take a closer look at the middle ear. So we're inside the ear. Here's the external acoustic meatus. This is the tympanic membrane, or the eardrum. And then on the medial side of that would be the tympanic cavity. That's that air-filled space. And these 3 tiny bones, the malleus, incus, and stapes, inside of the middle ear. And you can get just a glimpse of that auditory tube that connects the middle ear to the nasopharynx and allows for equalizing of pressure. Inside of the middle ear, there are 2 muscles that contract to restrict the movement of the ossicles when potentially damaging loud noises occur. So the tensor tympani, which is connected to the malleus over here, and the stapedius, which is the smallest skeletal muscle of the human body, attached to the stapes. And its job is to stabilize this smallest bone, the stapes. These function in your acoustic reflex. When there's a really loud sound, then these 2 muscles contract. So the stapedius and the tensor tympani contract in response to a loud sound. And that means that the sound doesn't continue into the inner ear and damage those really sensitive sensory structures of the inner ear. So if you hear a loud sound and then it feels like you're deafened for a while, it's because these 2 muscles are contracting. They're stopping the movement of the auditory ossicles. So it's stopping sound from entering into your inner ear, which is pretty cool. If we were to go into the inner ear, then we'd be going in through this oval window. So the stapes transmits vibrations from the incus, which transmits vibrations from the malleus, which is transmitting vibrations of the tympanic membrane. So when sound waves come into the ear, they cause the tympanic membrane to vibrate. That causes the malleus to vibrate, causes the incus to vibrate, causes the stapes to vibrate, and that stapes knocks on the oval window, which marks the boundary between the middle ear and the inner ear. So we go from a really big surface area of the tympanic membrane to a really small surface area of the stapedius and that oval window. And this is important because we're going from vibrations in air in the outer ear and the middle ear to vibrations in fluid inside of the inner ear. Fluid takes a lot more force to move. So that's why it's important that the stapes is a lot smaller than the tympanic membrane, because that amplifies those sound waves. So we're diving now into the inner ear. Illustrated over here in this schematic of the inner ear is the stapes, that tiniest bone of the human body. And number 2 right here would be the oval window, the opening into that fluid-filled inner ear. Inside of the inner ear are the structures of hearing and of equilibrium. It's 3 different senses and 3 different sensory structures. So this schematic is to help make everything a little bit less complicated. And I've posted this on Canvas for you if you want to check that out. So it's to give you an idea of these 3 different senses and different sensory structures of the inner ear, as well as the things that they all have in common. And that's what's illustrated up here in this basic version. So the things that they all have in common is that they're all surrounded by what's called a bony labyrinth. A labyrinth is sort of like a maze or a series of caves. And you can think about this bony labyrinth as a series of caves through the temporal bone. So there's openings in the temporal bone that form these structures. So number one is always going to be the temporal bone, but the bony labyrinth gets different names according to where it is. So the bony labyrinth, made up of structures of the temporal bone, it's filled with a fluid, so a space between 1 and 2, filled with the fluid. That fluid is perilymph. Then following all of the nooks and crannies of the bony labyrinth is a membranous labyrinth. So there's a membrane that's following the curves of the bones and lining those nooks and crannies. That's the membranous labyrinth. And inside of the membranous labyrinth is going to be a fluid that's endolymph. So there's a bone layer, then there's perilymph, then there's a membrane, and within that membrane, endolymph. Within the endolymph, for all three of these sensory structures, you're going to find a receptor epithelium. These are hair cells. So they have hair cells that project through the endolymph, oh no, and into an overlying gel mass. And so when either the membranous labyrinth moves or the overlying gel mass moves, then those hairs bend and they send signals down the vestibulocochlear nerve. So that's the general model, is that when these hair cells bend, then they're causing a sensory signal to be sent along the vestibulocochlear nerve. So these are mechanoreceptors. Your senses of hearing and of static and dynamic equilibrium are all mechanoreceptors. They're detecting a change in the shape of the cell. They're exteroreceptors because they're detecting something about the outside world, and they're considered a special sense. So I'll talk through the details of some of the specifics of these. But first I'll show you a little bit less cartoony, a little bit less schematic of where we are. So I said that we're within the petrous portion of the temporal bone, and that's what's illustrated here. So this is all the petrous portion, that raised section of the temporal bone. Out here would be the external acoustic meatus, and then right here where this nerve is leaving, that's the internal acoustic meatus. So within that raised portion, the temporal bone, that petrous portion, that's where we'll find the cochlea, that structure for hearing, as well as the semicircular canals and the vestibule. I like this illustration over here because it shows that, you know, sometimes when you see the cochlea illustrated like this, it looks like you're going to find just a tiny little snail shell somewhere inside of the brain, but really it's just tunnels through the temporal bone that take the shape of a snail shell. And over here, you can see the semicircular canals and the membranous labyrinth that lines them, and the vestibule would be down here. And out here would be the auditory ossicles of the middle ear. So we've located ourselves, we're within the temporal bone, within the petrous portion. And it's these tiny tunnels through the petrous portion of the temporal bone. So here we are again, looking in the temporal bone at some of these structures of the inner ear within the petrous portion of the temporal bone. These cavities within the bone make up the bony labyrinth. So all of these hollow spaces within the bone are the bony labyrinth. And within the bony labyrinth are fluid-filled tubes and spaces. And these fluid-filled tubes and spaces make up the membranous labyrinth. The receptors for equilibrium and for hearing are within the membranous labyrinth. Those receptor epitheliums are within the membranous labyrinth. The space between the bony labyrinth and the membranous labyrinth is filled with a fluid that's called perilymph, and the space inside of the membranous labyrinth contains that fluid that's called endolymph. So the bony labyrinth has 3 different regions. Over here is the cochlea. The cochlea is the bony labyrinth, and the membranous labyrinth is called the cochlear duct. So over in that snail shell looking thing, that's the cochlea, which houses the organ of hearing, which is called the spiral organ or the organ of Corti. And I'll go into detail about that in a second. The vestibule, this big open space right here, this big open chamber. The vestibule is the bony labyrinth, and then the membranous labyrinth is called the macula or the utricle and saccule. And the receptor epithelium is the macula. And that houses those organs of static equilibrium. And then lastly, these 1, 2, 3 semicircular ducts are the membranous labyrinth within the bony semicircular canals. And within this ampulla right here is the sensory epithelium. So we'll go through each one of those. So the first one is this big space, the vestibule right here. So if you were to, be inside of the middle ear, you're a little tiny person that was shrunk down like in Magic School Bus, and you climbed in through the oval window, you would find yourself in a big open space inside of the temporal bone. And that big open space is called the vestibule. So that's the bony labyrinth. And the membranous labyrinth within the vestibule is called the utricle and the saccule. And these utricle and the saccule, they're responsible for your static equilibrium. So equilibrium refers to the awareness of your head position, and static equilibrium is detecting the orientation of the head when the body is stationary, or linear acceleration of the head. So the utricle and the saccule is what lets you know when you're, for instance, going up in an elevator. You can feel that you're going up, even if you can't see any visual cues. That would be linear acceleration. Or if you have your head tilted forward, you can detect that. And that's static equilibrium. And all of that's going to be detected by these structures within the vestibule. So you can see in part A down here, the maculae are located within the walls of the utricle and the saccule. And these are these membranous sacs within the vestibule. If you took a little bit of a closer look, then what we can start to see is that receptor epithelium. That's the macula. And it has hair cells that have specialized cilia that extend up into this overlying gel mass. This overlying gel mass is called the otolithic membrane, meaning that it contains tiny crystals within it, tiny rocks. And these rocks are sitting in sort of a jello layer. When gravity acts on the otolithic membrane, then it causes the tiny hairs at the end of the hair cells to bend, and that sends messages down the vestibular branch of cranial nerve number 8. So because of their orientation in the head, the utricle is sensitive to changes in horizontal movement, and the saccule is giving information about vertical acceleration. And let's take a look at how that works. So here's the volunteer. She's got her head upright, and this is the receptor epithelium down here. That's the macula with the hair cells that are sticking up into an overlying gel mass, the otolithic membrane. Otolithic membrane is made up of a gelatinous matrix, and it has calcium carbonate crystals in it, these tiny rocks. And when gravity acts on these tiny rocks, then it causes the hair cells to be stimulated when the head moves. So here she's tilted her head downward. Gravitational force is acting on these tiny rocks. It's pulling the otolithic membrane, and that's causing these tiny hairs, these cilia of the hair cells to bend, and that sends a message along the vestibular nerve. So that's how you can detect whether you're moving in an elevator, if your head is tilting. All of that is static equilibrium detected by the macula within the utricle and saccule within the vestibule. Your next sense of equilibrium is dynamic equilibrium. Dynamic equilibrium is when your head is in motion. And this is detected by the Crista ampullae within the semicircular ducts, within the semicircular canals. So here's the vestibule again with the utricle and the saccule. And then there are three semicircular canals. That's the bony labyrinth. Within them, semicircular ducts. That's the membranous labyrinth. And at the base of each one of these semicircular ducts is an expanded area called the ampulla, which has the sensory receptor epithelium, that is the Crista ampullaris. So in this elevated region, you can see a close-up right here. There's endolymph, the fluid within the semicircular ducts. There's the Crista ampullaris. That's the receptor epithelium with the hair cells. And above it is the cupula. That's an overlying gelatinous dome, and it's surrounded by endolymph. And these receptors are going to be responsible for detecting the rotational movement of the head. They're all at 90-degree angles to each other, so they can detect shaking your head no, nodding your head yes, and spinning around in a circle. So here we are within the temporal bone, and those are those three semicircular canals of the bony labyrinth, all at right angles to each other, allowing you to detect dynamic equilibrium in three different planes. So what's going on within the bony labyrinth? Remember that within the bony labyrinth is a membranous labyrinth, and within the membranous labyrinth is that fluid, the endolymph. When you're turning your head, you're turning faster, and the fluid is staying in place due to inertia. So the fluid moves more slowly than your head does, and it causes movement of that fluid. So when the head is rotating, it causes the endolymph within the semicircular ducts to move, but more slowly than your head is. And what this does is when this fluid is flowing by the ampulla, these enlarged regions at the base, then it's causing the fluid to move, and it's pushing against the cupula. When the cupula bends, then that causes bending of the hairs in that receptor epithelium, the Crista ampullaris, and that's what causes a nerve impulse to be sent to the brain. So if you're rotating your head, as this volunteer is doing, then it's causing movement of the fluid in just one of the semicircular canals, and that's how you know that your head is turning in a particular direction. If you've ever drunk alcohol and gotten dizzy, or you feel like you got the spins, one reason that this might be occurring might have to do with the semicircular canals, these structures that detect dynamic equilibrium within the inner ear. So if somebody has drunk alcohol, what happens is that alcohol, which is less dense than most fluids in the body, less dense than water, might be diffusing into the endolymph, so that this endolymph now contains alcohol. And because that endolymph is now less dense than it previously was, it causes increased movement of the cupula. There's less pressure on the cupula, so it can move more freely. If it's moving more freely, what this means is that somebody who has alcohol in their endolymph has this erroneous sensitivity to linear accelerations. Basically, you think that everything is moving a lot more than it really is. this sensation is called the spins. If you turn your head a little, it feels like you've turned your head a lot because the cupula is moving a lot more freely. So that might be one reason why people get the spins when they overindulge in alcohol, is increased movement of the cupula and therefore increased signaling within the Crista ampullaris, within these semicircular canals. The last structure of the inner ear is the cochlea. That's the bony labyrinth. Inside of the cochlea, the membranous labyrinth is the cochlear duct, and that's illustrated in purple here. So this whole bony structure, that's the cochlea, the bony labyrinth, filled with paralymph. That fluid is shown in blue. Then the membranous labyrinth is the cochlear duct, filled with what's shown in purple, that's endolymph. And within the cochlear duct is that receptor epithelium. That receptor epithelium is called the spiral organ, or the organ of Corti, or sometimes the spiral organ of Corti. So I'm going to try to stick with spiral organ because you know that we're moving away from naming things after people. So the organ of Corti, or the spiral organ, is the receptor epithelium that contains hair cells. Within the cochlea, the overlying gel mass that's going to be responsible for bending those hair cells is called the tectorial membrane. So this is a section through the cochlea, that bony labyrinth that exhibits a snail spiral-like shape and has these fluid-filled ducts. Here's a close-up through it. So this whole thing is the bony labyrinth, the cochlea, and some portions of it contain paralymph. That's always going to be inside of the bony labyrinth. Within the bony labyrinth is also the membranous labyrinth. That's the cochlear duct that contains endolymph. And then the receptor epithelium, the spiral organ, and the overlying gel mass, that's the tectorial membrane. But let's look a little bit more closely at the cochlea and the spiral organ. So out here, the scala tympani, this is going to be filled with paralymph. That's part of that bony labyrinth. And then everything enclosed up here is going to be inside of the membranous labyrinth. So there's going to be endolymph inside of here, illustrated in purple. The receptor epithelium is these cells right here with their stereocilia sticking up. So these are mechanoreceptors. They're going to be responsible to changes in the shape of the cell. And just above it is that tectorial membrane, the overlying gel mass that's going to cause those hairs of the hair cells to bend. So I try to find a way to show how this was occurring. So what happens is we get sound waves that are coming in through the external ear, and they're impinging on the eardrum, causing it to vibrate. That causes the ossicles of the middle ear to vibrate and the stapes to move in and out of the oval window of the inner ear. When the stapes knocks on the oval window of the inner ear, then that causes the paralymph to move in the scala vestibuli. That's part of the bony labyrinth that's filled with paralymph. So we have paralymph moving in the scala vestibuli, and this causes physical waves that move up and down the scala vestibuli of the cochlea. So short waves, high frequency or high pitch, are acting at the base of that spiraling cochlea, and long waves are acting at the apex of the cochlea. And these waves are causing changes in the membrane of the membranous labyrinth and causing distortion at the organ of Corti, so causing movement of the tectorial membrane or of the organ of Corti along the basilar membrane, and that causes the hair cells to bend So these hair cells that are in contact with the tectorial membrane, these mechanoreceptors pass impulses along the cochlear branch of the vestibulocochlear nerve. So the sound wave continues through the scala vestibuli and then back down through the scala tympani and exits out the round window, so letting those sound waves back out of the inner ear. So let's take a look at this sound path through the cochlea. So step one, you're getting sound waves that are entering into the inner ear, causing that tympanic membrane to vibrate. The tympanic membrane vibration moves the auditory ossicles and amplifies those sound waves. So up here, the amplitude of the sound waves at the external ear, and then they're getting even bigger in the middle ear. Remember, I said that's going to be important because after the middle ear, sound waves are no longer traveling through air, they're traveling through fluid. So it's important that they get amplified here. The stapes knocking at that oval window is generating pressure waves in the paralymph within the scala vestibuli. That's the compartment of the bony labyrinth that contains paralymph. And those pressure waves travel along the scala vestibuli, and they're traveling along the scala vestibuli. And if they're really short waves, then they're acting here at the base of the cochlea. And if they're really high-pitched waves, they're acting up here at the top of the spiral of the cochlea. So this one looks like it's about a mid-frequency pitch that's causing this part of the vestibular membrane to vibrate. And these vibrations cause pressure wave formation to occur within the endolymph, within the cochlear duct, that's the membranous labyrinth, and it causes displacement of this very specific region of the basilar membrane. Hair cells that are in the spiral organ, or the organ of Corti, get distorted because of that overlying tectoral membrane. And that sends a nerve impulse along the cochlear branch of cranial nerve number 8. Any sound waves that are remaining, any pressure waves that are remaining, travel all the way up to the top of the cochlea and all the way back down through the scala tympani. They're traveling out and they're exiting out the round window. This is a lot of details for the sound path through the cochlea. You should know that sound waves are coming in through the external acoustic meatus, vibrating the tympanic membrane, vibrating the auditory ossicles, and getting amplified at the auditory ossicles. Then sound waves are traveling through the paralymph of the scala vestibuli and traveling into the cochlear duct that is the membranous labyrinth and causing distortion in the basilar membrane, causing that organ of Corti to move and be stimulated when the hair cells that stick up into the tectoral membrane are bent. For your test, you might also study an image like this one over here, because I might ask you a question, like I would show you this image unlabeled and ask you, what is this structure? And you would tell me it's the tectoral membrane, or it is the overlying gel mass. I might ask you what fluid is contained within this compartment. You'd tell me it's the endolymph. I might ask you what fluid is contained in this compartment. You'd tell me it's the paralymph. So things, identifying structures like that, as well as identifying structures of the external ear, of the middle ear, and things like the auditory tube. So special senses are a lot of fun, but it can also be challenging. So please let me know if you have any questions.